A likely substrate of depressive disorders is disrupted synaptic plasticity, at the core of which is cytoskeletal proteins and microtubule activity. Alpha-tubulins undergo post-translational modifications, or PTMs, which affect growth and rapid reassembly. Tyrosination results in more dynamic microtubules and increased plasticity, while detyrosination, acetylation, and deglutamylation may represent a decrease in microtubule dynamics and therefore reduced plasticity. Examples of alpha-tubulin PTMs are visible in Figure 1. Preclinical studies indicate plasma and cerebrospinal fluid levels of alpha-tubule PTMs as reflective of changes in the brain. In this study, we investigated whether alpha-tubulin PTMs are detectable in human samples and whether these modifications could serve as potential biomarkers of neuroplasticity in correlation with Hamilton Clinical Depression scores. Human blood samples were collected from patients with major depressive disorder and healthy age-matched controls using venipuncture. Alpha-tubulin PTMs were quantified using infrared Western blot. The two ratios of interest in this study were the amount of tyrosinated tubulins relative to detyrosinated and acetylation relative to total alpha-tubulin concentration. In a healthy brain, a high tyrosinated-detyrosinated ratio and a low acetylation relative to total tubulin ratio is suggestive of dynamic microtubules and therefore typical neuroplasticity. We found significantly decreased tyrosination relative to detyrosinated tubules in moderate and severe depressed patients. This can be seen in Figure 2. Sex was not a significant factor here, as ratios did not statistically differ between male and female plasma, as seen in Figure 5. Finally, lower tyrosinated-detyrosinated ratios correlated with higher Hamilton clinical scores. This can be seen in Figure 4. In conclusion, the current study demonstrated that alpha-tubulin PTMs are detectable and quantifiable in human plasma, and that the ratio of tyrosinated tubulins is decreased in depressed samples. This finding indicates decreased microtubule dynamics, which correlates with clinical ratings of depression. Alpha-tubulin post-translational modifications may therefore be a plasma biomarker of depression, which may be used for monitoring of drug response in clinical trials. Thank you.